Welcome to the Mitosis Notes for Biology. This is Miss Mueller featuring Miss Bao to teach you about mitosis. If you recall, DNA stands for deoxyribonucleic acid. So what macromolecule is that? Your options are carbs, lipids, proteins, or nucleic acid. What do you think it is? You're right. Nucleic acids. The function of this is to contain the code for life, meaning that it's the pattern for making proteins, which is extremely important. So think of DNA as like an IKEA manual, and the proteins are all the parts. You actually have to read the manual in order to make the parts work, hence proteins. So DNA is found in the nucleus. If you look at this electron micrograph here, you can see that this really dark spot is the nucleus. So its characteristics is that it is a double-stranded helix, which is a really fancy way of saying twisted ladder. So if you look, here's a ladder, here's a twisted ladder, that's what it looks like. No joke. We didn't know this until the 1950s when Watson and Crick did their famous experiment, so it's pretty cool. So DNA can take on a few different forms. It's not always just this twisted ladder business, okay? So chromatin is what we call an uncondensed form of DNA. So condensed means close together. Think about condensed soup, okay? When you make condensed tomato soup from Campbell's, you have to add water or milk to it. Uncondensed means that it's really loosey-goosey and relaxed. So how I like to think about it is look at these electrical cords, okay? They're all like messy and they're all knotted up and they take up a lot of space. So the DNA is loose. Sometimes DNA looks like a chromosome, which is the condensed form, meaning that it's really tightly wound and it's very organized. Hence, looking at our extension cords like when you are storing them when they are not in use in your garage. So it's organized. So here's a big picture. Chromatin looks like a big crumbled up mess of yarn or rope or Christmas lights depending on the time of year. And here, a chromosome is the nicely wound and organized form of DNA. So when the cell divides, it must be copied. Think about a baby. We want our baby to have a brain. We want them to have lungs. We want them to have a heart. So everything in that baby that needs it to survive, you need to copy that, Mom, when you're pregos. This is known as replication. So in order for your cell to divide, you're going to make a new copy of DNA for that new cell. So when the cell divides, you're going to make this new DNA through DNA replication, and then the rest of the cell is going to follow along in something called mitosis. So this picture is what the cell goes through called the cell cycle. So here we have interphase, right here, which, if this image is to scale, which it is, it takes up the majority of the cell's life. We have cell growth in G1, S phase, where the DNA is copied. S stands for synthesis, fun fact, and G2, which is growth and preparation. All the organelles are being copied, the cell gets bigger, it's just ready to make another baby cell. Then we have M phase, which is part of the cell division phase. Mitosis is what M phase means, which is formally defined as division of the nucleus, and cytokinesis, which is the division of the cytoplasm or the rest of the cell. Once that occurs, then it's going to go through the cycle again and make more cells. So here is an image that I drew, because I find that sometimes when you look at images online, you're like, how do I draw this? Well, here it is. I drew it for you right here. So here, G1, S, G2, that makes up interphase, which is when the cell grows and develops, mitosis, and cytokinesis. So interphase. Most of the time is spent in this phase. G1 stands for growth. S is for DNA synthesis, where the DNA is copied. G2 is when the cell prepares for division. So sometimes cells exit the cycle in this thing called G sub 0, where they simply exist and never divide. Your brain cells are an example of this. If you've ever heard of like you use it, you lose it, or you're burning brain cells, sometimes when you have brain damage, it never goes back to normal, meaning that a lot of the cells are in G0. There is some evidence that our brain cells can go back into mitosis and the cell cycle, but again, for the most part, they stay in G0. So if you think about it, think about a cell that maybe would not want to be in G0. Think about a cell that's always replicating or that you're always going to need new cells. Think about it. Pause the video. Think about it. Now that you've paused the video, think about our skin. How often do you brush up against something and hurt yourself? What about your intestines? What about your colon? Think about it. So here's a picture of a cell. Sorry, it's kind of blurry. Here is a cell in interphase where I have this chromatin, or the DNA inside the cell that's all loosey-goosey and unraveled within the nucleus. Then there's these things called centrosomes, which are often called centrioles, that are just hanging out in the cell. I'll get to them later. So mitosis is the division of the nucleus. Okay, so that is going to occur when the cell begins to divide to make a new cell. There are 4 phases: prophase, metaphase, anaphase, and telophase. A way to remember this is PMAT, or PMAT. During prophase, the chromatin is going to condense to become a chromosome, meaning you have a big old tangled mess going to a neatly organized non-mess. The nuclear membrane is going to disappear, that's being shown here. And the centrioles are going to move to opposite ends of the cell. These centrioles make spindle fibers, which are these little fiber things right here. Think of those as fishing line, and the centrioles are the fishermen. So these things are made up of something called microtubules, which is a protein. So here we go, macromolecules. You're welcome. So in your notes, this is what I would draw. This is prophase. Here I have my cell. Here I have the centrioles with spindle fibers on opposite ends of the cell. Here I have the nucleus that is disappearing, and here I have my nice chromosomes that are no longer a bundled up mess. Metaphase is the next phase, and chromosomes are going to line up along the center of the cell. Meta means middle. M, think middle. So the spindle fibers are going to attach the centromere to move them to opposite ends of the cell. Oopsie. So a centromere is right here. If you're lucky, Miss Bao, me, or Miss Kingman will teach you how to do the butterfly dance. If you think about a chromosome, think of it like a butterfly. Okay, here's one wing, here's the other one, here is the butterfly body. So here's how I would draw this in your notes. So metaphase, the chromosomes, are lined up along the middle. The spindle fibers are attaching to the center of the cell called the centromere. I probably should have labeled that, but you could do that in your notes. The centrioles are the things that are going to be attaching the spindle fibers to the centromere. Okay? So again, centromere, butterfly body, right here, separating the left and right sides of those chromosomes. Anaphase is the next phase. The sister chromatids are going to be separating at the centromere. Okay, so boom, separating from boom. The chromatid is 1/2 of a chromosome. So this is a chromatid and this is a chromatid. So they will be slowly pulled to opposite ends of the cell. Okay, if you think about it like a butterfly, what are we doing to the butterfly? Can you picture it? Yeah, we're going to rip the butterfly wings off. Don't worry, you'll learn the butterfly dance. It'll be cool. Get excited. So draw this in your notes. Okay, so here I have my sister chromatids being pulled apart. Here I have an oopsie poopsie. I should not have this word pointing to this right here. That is not a centriole. This is a centriole. What should be here? You're right, chromatid. So again, here I have chromatid and a chromatid being held together by the centromere. They will eventually be separated into left and right halves. So again, anaphase, I want you to think of away. They're being pulled away from each other. Telophase is the last phase of mitosis. The spindle fibers are going to disassemble, meaning they're going to go away. The chromosomes are going to return to being a crumbled up mess. Think of them as the relaxed form, okay? They get really messy again. The nuclear membrane is going to reform. Okay, so we're going to reform that nucleus that's going to keep the DNA safe and away from damage. So here, I have a picture. Okay, we're going to be separating right here in the next part of the cell division cycle, which is called cytokinesis. My nuclear membrane is going to reform, and I have my chromatin in both cells. Cytokinesis is not a part of mitosis. It is separate because it is division of the cytoplasm. Cytoplasm, remember, is the jelly-like substance that's within your cells that suspends all of your organelles or cell parts. So animal cells often pinch together to create something called a cleavage furrow, where plant cells develop a cell plate. All I want you to appreciate is that different cells have different ways of doing cytokinesis, but the end of the story is that the cytoplasm is going to divide, so I get two separate cells. So here I have telophase right here. Right here it's going to pinch off, and here I have my two cells that are identical to each other. We call these daughter cells. I hope you enjoyed the show.